Welcome to the deep dive. If you've ever wondered what's actually going on, deep inside our cells when things go wrong, when a cell is literally fighting for its life, then this deep dive is for you. Today, we're diving into the really fundamental rules of how cells survive, or don't, the mechanisms of cell injury. Think about it. A cell gets hit with stress, maybe a toxin, maybe it's starved of oxygen, maybe radiation, and it has to make this huge decision. Does it try to fix things, adapt, or is it time to self-destruct?
And that decision, it's incredibly complex. It really hinges on the specific type of cell, what state it's in at that moment, and crucially, how adaptable it is. Just think about, say, a skeletal muscle cell versus one in your heart.
Right, like you can strain your arm muscle, rest it, maybe restrict blood flow for a bit, and it can often recover, it can handle, a temporary shortage.
Exactly, but a cardiac muscle cell, the heart never rests. Its energy demand is constant, and it's incredibly high. That makes heart cells way more vulnerable to hypoxia, lack of oxygen, than almost any other tissue. If the same stress hits, that heart cell is in trouble much, much faster.
So the stakes are definitely high. And our sources make it really clear cell injury isn't some neat step-by-step process. It's messy. It's like a whole bunch of interconnected systems starting to fail, sometimes all at once.
Yeah, it's a cascade.
So our mission today is to kind of map out those main failure points inside the cell, understand how we get from that initial stress to the end result, whether that's, the chaotic breakdown of necrosis or the more orderly self-destruction of apoptosis.
Okay, let's lay out the battlefield then. There are basically four main cellular targets that these damaging stimuli go after almost immediately. The mitochondria, the powerhouses, the cell membranes, the walls, the machinery for protein synthesis and secretion, and of course the DNA, the blueprint.
And we absolutely have to start with the energy, right? The mitochondria. In almost any crisis, the first thing you worry about is the power going out.
That's a perfect way to put it. Mitochondria are definitely the cell's power plant, but they're also kind of the arbiters of life and death. They're super sensitive to almost any kind of injury, but especially to lack of oxygen, hypoxia. The immediate problem is ATP depletion. ATP is that energy currency the cell uses for basically everything. When oxygen levels drop, the main ATP production line, oxidative phosphorylation, stalls.
So what happens when the cell starts running out of cash, essentially? What's the first critical thing that stops working when ATP levels fall drastically, say, down to only 5 or 10%?
Well, the first major casualty is often the cell's structural integrity. Specifically, this crucial pump called the NAM plus K plus ATPase, the sodium-potassium pump. It uses a ton of energy to constantly pump sodium out of the cell.
Ah, okay. So if the pump fails because there's no energy.
Sodium rushes in, and water always follows sodium osmotically.
So the cell just starts to swell up, like a balloon filling with water.
Precisely. You get this acute swelling of the cell itself, and also the organelles inside, like the endoplasmic reticulum, starts to dilate. Now, if the oxygen supply is really cut off, like a ischemia-reduced blood flow, the cell tries this desperate backup plan.
What is it?
It switches to anaerobic glycolysis, trying to make ATP without oxygen.
Sounds like kicking on the emergency generator.
It is, but it's really inefficient. It burns through the cell's stored glycogen incredibly fast, and worse, the main byproduct is lactic acid.
Right, I've heard of that.
And that lactic acid builds up, causing the inside of the cell to become acidic. The pH drops, and that acidic environment itself starts to shut down lots of other essential enzymes. It makes everything worse.
Okay, so the cell is swelling, it's becoming acidic, it's running out of fuel. Is there a specific point where it just can't come back? A point of no return for the mitochondria? Our sources talk about something called the mitochondrial permeability transition pore, the MPTP. What's that?
Think of it like a catastrophic breach in the mitochondrial wall. It's a large channel that opens up in the inner mitochondrial membrane. When the MPTP forms and opens, it causes a complete collapse of the mitochondrial membrane potential. Meaning oxidative phosphorylation stops dead. No more ATP production from that mitochondrion. It's a fatal blow. The opening of that pore essentially signals irreversible damage and commits the cell to necrosis. Once that gate opens, the fight is basically over for that cell.
Wow. Okay, the power plant is permanently offline. Let's move to the second major target you mentioned, calcium homeostasis. Normally, calcium is carefully controlled, right? But here it becomes a problem.
That's exactly right. Normally, the concentration of free calcium floating around in the cytosol is kept incredibly low. But injury, especially from ischemia or toxins, disrupts this tight control.
Oh.
First, calcium gets released from the cell's internal storage tanks, the ER, and even the mitochondria themselves. Then the damaged cell membrane might let more calcium leak in from the outside, so you get this sudden massive spike in cytosolic calcium.
And why is a flood of calcium so bad?
Because this excess calcium acts like a master switch, activating a whole bunch of destructive enzymes that are normally kept in check.
Like what kind of enzymes?
Well, you get activation of phospholipases, which start chewing up the cell's membranes. Then there are proteases, which break down not just membrane proteins, but also the cell's internal scaffolding, the cytoskeleton. You also activate endonucleases, which begin chopping up the DNA and chromatin. And sort of ironically, it even activates ATPases, which burn through whatever little ATP might be left even faster.
So the calcium overload basically triggers the cell's own demolition toolkit against itself.
Precisely. It unleashes this cascade of self-destruction.
Okay, so we had the energy crisis with mitochondria, the activation of damaging enzymes by calcium. The third failure you listed was membrane damage. This sounds like the actual structural collapse. What causes the membranes to fail?
It's often a combination of hits. First, those calcium-activated phospholipases we just talked about are actively breaking down membrane phospholipids. Second, because ATP is low, the cell can't synthesize new phospholipids to repair the damage.
A double whammy.
Exactly. And third, you have damage from those reactive oxygen species, or ROS, we'll talk more about them in a second, which cause something called lipid peroxidation, basically making the membrane lipids rancid and unstable.
And is the main problem here the cell just bursting open? Or is it more about what leaks out or in?
It's definitely about the leakage. Damage to the main plasma membrane means the cell loses its osmotic balance, more water might rush in, but it also means vital molecules like cofactors needed for metabolism leak out, which makes the ATP depletion even worse.
Okay.
But maybe the most critical membrane failure is damage to the lysosomal membranes. Lysosomes are like the cell's recycling center, but they're filled with really powerful digestive enzymes. Acid hydrolases like proteases, DNases, RNases.
And if those leak out?
If they leak into the cytoplasm, remember how the injured cell is often acidic because of that lactic acid buildup?
Yeah.
Well, these lysosomal enzymes are designed to work best in an acidic environment. So they leak out into the now acidic cytoplasm, become highly active, and just start dissolving proteins, RNA, DNA, the works. It dramatically accelerates the whole process of necrosis, that messy, uncontrolled cell death.
Okay, that sounds pretty catastrophic. So far, we've mostly talked about pathways leading to this messy collapse and necrosis, but cells can also undergo a more programmed death, right? Apoptosis. This often involves reactive oxygen species, or ROS.
Yes, ROS. These are unstable molecules, usually derived from oxygen, that have an unpaired electron. This makes them highly reactive. Think of like tiny bits of shrapnel bouncing around inside the cell, damaging whatever they hit.
Are they always bad?
No, actually, cells produce some ROS normally, especially during metabolism in the mitochondria. And the cell has defense systems to handle them. The problem arises when there's an imbalance, either too much ROS production or the defenses are overwhelmed. That state is called oxidative stress.
Where does this excess ROS come from during injury?
It can come from several places. Problems in the mitochondria during ATP production, exposure to radiation like X-rays, inflammation is a big source. White blood cells actually produce bursts of ROS to kill microbes. Also, the breakdown of certain toxins or drugs can generate them.
And the cell's defenses, what are they?
The cell has a pretty good toolkit. There are antioxidants like vitamins E and A and glutathione. There are proteins that bind up metals like iron and copper, which can catalyze ROS formation. And then there are specific enzymes dedicated to neutralizing ROS, like catalase, superoxide dismutases, SODs, and glutathione peroxidase.
But if these defenses fail, what does the ROS shrapnel actually damage?
Three main things. First, lipid peroxidation. They attack the lipids in cell membranes, setting off a chain reaction that damages the membrane integrity. I mentioned that earlier. Second, protein modification. They can damage amino acids, cross-link proteins, mess up enzyme function, and damage structural proteins. And third, lesions in DNA. They can cause breaks in the DNA strands or modify the bases.
Which logically leads us to the third mechanism, DNA damage. If ROS or radiation or even just aging causes significant damage to the cell's blueprint, the DNA, the cell has a plan for that, doesn't it?
It absolutely does. It involves a really famous protein called P53, often called the guardian of the genome.
How does it work?
When significant DNA damage is detected, P53 gets activated. Its first job is to halt the cell cycle, basically. Hit the pause button. It gives the cell time to try and repair the DNA damage.
Okay, so it tries to fix it first.
Yes. But if the damage is too severe, too extensive to be repaired accurately, P53 then switches roles. It initiates apoptosis, programmed cell death, usually by triggering the mitochondrial pathway we discussed earlier.
So the cell essentially decides to commit suicide rather than risk becoming cancerous with damaged DNA.
That's exactly the idea. It's a crucial anti-cancer mechanism. If a cell survives with damaged DNA, it could lead to mutations that cause uncontrolled growth, which is cancer. So P53 forces the cell to die for the greater good of the organism.
That's incredible quality control. And speaking of quality control, there's another system failure that can trigger death, this time in the endoplasmic reticulum, related to proteins not folding correctly.
Right, that's ER stress and the unfolded protein response, or UPR. The ER is like the cell's factory for folding and modifying many proteins. Sometimes due to genetic mutations, infections, or stresses like hypoxia, proteins don't fold correctly. They misfold and start to accumulate in the ER.
So the cell tries to fix this too.
It does. The UPR is initially an adaptive response. The cell tries to cope by, for example, making more chaperone proteins to help with folding, and temporarily slowing down overall protein production to reduce the load.
But what if that's not enough?
If the problem persists, if the ER gets overwhelmed with misfolded proteins, that's what we call ER stress. The UPR machinery shifts gears. It stops trying to adapt and instead actively triggers apoptosis signals, activating enzymes called caspases that carry out the programmed death sentence.
And this failure of protein folding is actually behind quite a few major diseases, isn't it?
A huge number. The accumulation of misfolded proteins and the resulting ER stress is now known to be a key factor in many neurodegenerative diseases. Think Alzheimer's, Parkinson's, Huntington's, prion diseases. It's also implicated in things like cystic fibrosis and even type 2 diabetes. It really highlights how fundamental this cellular quality control is.
Okay, one last mechanism to touch on, which seems a bit different. Autophagy.
Autophagy literally means self-eating. It's a process where the cell essentially recycles its own components. It engulfs old or damaged organelles or clumps of protein inside a membrane bubble called an autophagosome, and then fuses that bubble with a lysosome so the contents get digested and the building blocks can be reused. It's primarily a survival mechanism, especially important during nutrient starvation. The cell cannibalizes non-essential parts to stay alive.
But this self-eating can also go wrong or contribute to disease?
Yes, the regulation of autophagy is really complex and context-dependent. Sometimes, like in cancer, autophagy might help tumor cells survive under stress. Other times, activating autophagy might help kill cancer cells. Its dysregulation is also linked to neurodegenerative diseases, maybe failure to clear out toxic protein aggregates and even inflammatory conditions. For instance, variations in an autophagy-related gene, ATG16L1, are linked to Crohn's disease, suggesting problems with this internal recycling can contribute to chronic inflammation.
Wow. Okay, what a journey through the cell's internal crisis management. We went from that immediate power failure, the ATP depletion, and the calcium becoming a weapon, through the walls crumbling with membrane damage, and then into these really sophisticated responses like oxidative stress, the P53 guardian, ER stress, and even self-eating with autophagy, all deciding whether the cell lives or dies.
Yeah, the key thing to remember is that cell injury isn't just one thing, it's a spectrum. On one end, you have reversible injury, where the stress is removed in time or the cell adapts and it recovers. But if the damage is too severe or goes on too long, it crosses a threshold into irreversible injury.
And that leads to death.
And that leads to death primarily through one of two main routes. Necrosis, which is that messy, uncontrolled inflammatory death caused by loss of membrane integrity and enzyme leakage, or apoptosis, the more orderly, programmed, non-inflammatory suicide.
And really understanding these molecular details, these failure points, the calcium influx, the lactic acid, the ROS, the misfolded proteins, that's not just academic, right? It's the core of understanding disease. Knowing why the cell is failing helps figure out how to intervene.
Absolutely. It's the foundation of pathology and ultimately therapeutics. It's trying to fix the root biochemical problem, not just manage the downstream symptoms.
So wrapping up, you've given us a lot to think about regarding how cells break down.
Well, here's one final thought. Building on that mitochondrial point of no return we discussed, we talked about the opening of that mitochondrial permeability transition pore, the MPTP, being this kind of final fatal step committing the cell to necrosis. So what if? What if future therapies could focus specifically on stabilizing that mitochondrial membrane, finding ways to keep that specific pore, that MPTP gate, shut even under stress? Could that potentially buy the cell precious time, maybe enough time to repair other damage and survive? Could we, in effect, make the irreversible? Reversible, at least for a while.